Now, with that, let me introduce the speaker of the day. Welcome, Dr. Pavan Kumar. Dr. Pavan Kumar is currently working as a senior scientist in Fish Genetics and Biotechnology Division, ICAR CIFE Mumbai. He has 15 years of experience in teaching and research related to DNA markers. He has published 90 research papers in different national and international peer-reviewed journals. He has discovered 4 new species, 1 ray fish, 2 telocid, and 1 parasite. Today's talk will cover the basic concept and evolution of DNA markers, DNA marker application in fish taxonomy, stock characterization, stock characterization, and to prevent illegal fishing. Welcome again, Dr. Pavan Kumar, and I'm passing the baton to you.
Thank you. Thank you, Dr. Pratish. Thanks for your nice introduction. And also, once again, thanks to the AFS team for giving me an opportunity to share my ideas and thoughts on this topic. So I'm sharing my screen. Yeah, since my bandwidth is a little bit slow, I am stopping my video, but the screens will be shared. Okay, thank you.
Thank you.
Yeah, so coming to the today's topic, that is application of molecular markers for sustainable fisheries. As we all are know that life is not so easy. So if you see the previous 2-3 years, so we were in a big trouble because of the new pandemic. And this is not new. If you see, if we look at the history, so since the beginning of the human culture and human evolution, there were so many pandemics were there, and which have challenged the survival of the humankind. But however, the humankind has, we can say the humankind has faced those challenges, and what happened to that? So you can see that from the Antonic plague and bubonic plague and smallpox, and now the even recent COVID-19. So each and every virus has a very devastating effect on the humankind, and it causes so many human loss. So did they really succeed in eradicating the humankind or not? So before going to the answer of this question, if we see at a broad basic concept and broad perspective, the most amazing thing in the universe, and even this is a night time in India, you can look at the sky and you can wonder of the stars and the moon and other matter. So if we look at the most amazing thing in the universe, so different persons have different perception of the universe. So probably the mathematician, the abstract concept of number would be the most amazing thing. Whereas for the physicist, so the matter, the constituent of the matter, and also the black holes are the most amazing thing, where the physical laws will be altogether different. So whereas for the chemist, the interaction of the molecules and how they are talking, and when they are interacting among themselves, what kind of products they are giving, and how the molecules are exchanging. So that would be very amazing. But above all these things are possible by a chemical entity which could harness the energy and replicate its own self, that is a life. So for biologists, the most amazing thing in the universe is life. So life has evolved into different forms, and it acquired all ecological niches, and it has caused a huge biodiversity. We will see one by one. If we look at the prehistoric era, the life has evolved, the life has originated in the aquatic environment. So the molecules, the atoms come together and they join into the macromolecules and the polymers, and these polymers got the energy and the capacity to replicate their own self. And through this process, the life has originated in the aquatic environment. After that, what happened to this life? So after that, as we know that if we come out of our comfort zone, then only we can realize our real potential. In the same way, the life also came out of its comfort zone and it occupied all the ecological niches. While it is occupying the all ecological niches, the conditions were not so conducive, not so favorable. However, more challenges will really bring out the potential of that particular entity. So while establishing in different ecological systems. So it has acquired certain traits and it occupied all the ecological niches of the earth, and it resulted in a huge biodiversity. And this biodiversity, if we look at this biodiversity, and each biodiversity has a unique contribution to the humankind. And before that, what is the molecular basis of life? So the molecular basis of the life is in the form of a nucleic acids. Someone says it is RNA, someone says it is DNA. So we can call it as a nucleic acids, the macromolecules, which consist of mainly carbon, hydrogen, oxygen, nitrogen, and phosphorus molecules. And this nucleic acid, here we can say it is a DNA. So the DNA is the blueprint of the life, and it is located in the nucleus. If you look at the cell at the resting phase, we can see the condensed form of the DNA that is in the form of the chromosomes. So the DNA has the genetic information, and that genetic information will be transcribed into messenger RNA. So these messenger RNAs are the molecules which carries the information for amino acid synthesis, and these amino acid synthesis this is the messenger RNA molecules, and the messenger RNA molecules carries the amino acid sequence information, and they will be translated into the proteins, and the proteins finally determines the phenotype, the structure of the appearance of the fish or appearance of the individual. So by looking at this, we can say the phenotype is determined by the genotype, that is the genetic constitution of the organism as well as the environment. The environment has a very important role in determining the phenotype along with the genetic composition of the individual. So, and if we see the mechanism of evolution, as I said, the first question, so though there are so many pandemics have invaded, have attacked on humankind, why the entire humankind has not been wiped out? So this is because of the variation. So the variation at the nucleic acid, at the DNA level, so the variation at the DNA level would lead to, suppose if the variation at the DNA level would lead to production of the new proteins, and these proteins would give an advantage to that individual. So that individual might be having the antibodies against to that particular pathogen. And because of that variation, the life will never going to extinct, especially for biotic factors. If there are any abiotic factors like any asteroid collision or any other things, so abiotic factors we could not say, but when it comes to the biotic factors, so most of the genetic variation protects the life to go into the extinct. So if we see the mechanism of evolution, what is the actual mechanism through which the life has evolved? So the most important thing is the mutation. So mutation means a nucleotide change in the DNA. So that is, there are different, based upon the kind of nucleotide substitution, there are different mutations are there. So these mutations happens during DNA replication stage, and DNA replications happens during the gametogenesis, and during the gametogenesis, because of the mitosis and the subsequent meiosis, there will be there is a possibility that mutations will happen, and along with that, the so here you can see, imagine these two circles representing two populations. So here the fish which is represented in the red color. So these fish may be moving from one population to another population. That means the genes are flowing from one population to another population. So subsequently, these populations in this population, the allele frequency would change, and it causes the changes in the phenotype and subsequently the variation. And also genetic drift. Here you have the equal number of blue color fish and red color fish, that means two different phenotypes or two different genotypes. But if some selectively some of the fishes are catching, so what will happen? Subsequent generations, so some of the fishes will be diminished. So that is the, so those fish will lose the contribution of their gametes to the next generation. So here you can see here the only the red color fish, this phenotype is there, whereas the blue phenotype has lost its chance to transfer gametes to the next generation. So and the last one is the natural selection. So suppose here this phenotype is particularly susceptible to prey by birds. So what will happen? So over the generations, this phenotype will lost from this population, and other phenotype will increase in the size, increase in the numbers. So these four mechanisms of the evolution, that is the mutation. This is the basic process for creating the nucleotide variation at the population, and when followed by the migration or genetic drift or natural selection. So these methods, these mechanisms play an important role in how individuals evolve over the time. And the individuals means it is always a population. So how populations evolves over a time, and how a new species will evolve, or how a new populations will be emerged. So as I said that the point mutations during the gametogenesis in the case of spermatogenesis and oogenesis. So during DNA replications takes place, that time that nucleotides will be substituted or it will be changed, and the sperm or the eggs, they may be having the mutations compared to their progeny, compared to their parent cells. So this kind of mutations is actually the rate of change is slow because of the selection pressure in some of the cases. So if this is a point mutations, we can call it as a transitions and transversions and indels and insertions and deletions. Some of the nucleotides may be inserted in the data DNA. Some of the nucleotides may be deleted from the data DNA. So this is one of the basic mechanism how variation will be created in the process of DNA replication, during the process of DNA replication. And also sometimes the chromosomal aberrations, there may be a certain portion of the chromosome may be deleted, or some maybe it may be inverse, or it may be ligated and inserted in another place. So these kind of chromosomal aberrations also create the variation at the DNA level. The rate of change is slow to moderate because they have high selections, severe selection pressure. So these kind of variations, the life cannot allow them to take place because it will cause a deleterious protein, or it may cause the it may create a hamp, or it may create a hindrance in the biochemical process, so that the individual may not able to survive. And also apart from this, there are certain regions which are repetitive in nature. So these repetitive in the nature, so these kind of variations is created during the replication process because of the slippage of DNA polymerase. So these kind of mutations will also happen. So the rate of change is high because they may not code for any protein. So since it is a non-coding protein, so the rate of change could be moderately high compared to other point mutations and chromosomal inversions or deletions. And transposable elements. So these transposable elements also causes genetic variation at the DNA level. There are two kinds of transposable elements. One is the cut and paste mechanism and copy and paste mechanism. So these nucleotides, they will excise themselves from the genome, and they will integrate at some other location of the genome. So that they will create the novel combinations, and they will create some kind of novel alleles. So this kind of rate of change also moderate to high because
Dr. Pavan, are you there? Seems like we are not hearing you.
And the other chromosomal aberrations and the transposable elements. So these are the primary factors that are responsible for the variation at the DNA level. And along with that, the migrations and the genetic drift and natural selections and these other factors will also determine the evolutionary trajectory of the population, how it will be evolved, and which alleles number will be increased, and which species will be emerged, and which species could be extinct. So if we look at the tree of life, so the common ancestor of all this life has evolved here somewhere, has originated here somewhere. So the population has slowly evolved over the time. So as the environment is changing, so the population also acquired certain mutations and all these factors are as what I said. So and because of those factors, the life has been so the life has been evolved into different distinct lineages, and we are here. So the humans are here, and along with the other mammals. So this is a broad phylogenetic tree of life. So if you see that here, there will be some other separate branches will be there. So in one way, we all are related. The humans are not an allied form of the life. We are also a part of the other life. However, we have evolutionarily, we have followed a different trajectory, and our neuron system has evolved in such a way that we started the critical thinking and critical question asking capacities. So here, broadly, I want to say that always the evolution takes place in a population, and during the evolution means the change. Change means the nucleotide allele frequencies change. Alleles means different nucleotides, different different DNA sequences. So when the DNA is changing over the time, so the alleles will increase their frequency, and that frequency, because of that allele frequency changes, it could lead to the establishment of that fixation of the alleles, and the populations over the time it acquired the new traits, and it could evolve as a new species. So if you look at the pyramid stage, biological pyramid, at the base level, the genetic diversity is very important, based on which the life will evolve on its own. And this is within the species, the genetic diversity. And after the second level, the species diversity, how many species are there? And after that, the ecosystem diversity. So when this pyramid is fulfilling all these different fractions, then the ecobiome will be healthy. So coming to the genetic marker, what is actually genetic marker? If you imagine this is the chromosome, so the condensed form of the DNA, this is the locus in the genetic terminologies. In the locus, we have we define the locus as the position of the gene on a chromosome. So in this locus, we have two alleles. You can see here, this is capital G and small g. So the capital G is a gene that may be coding for the enzyme G, and the small g is a gene that is coding for the alternative form of the gene, which could be responsible for a different color, eye color. So here, the allelic difference, this difference, either we could observe in the form of the morphology, or at the level of morphogyne, or sorry, at the level of enzyme, or at the level of the DNA sequence itself. So these kind of markers, we can call it as a genetic marker. So genetic marker, if we are observing any morphological trait and we are correlating that morphological difference with the genetic basis, so that we can call it as a genetic marker. If we are observing any enzyme differences and we are correlating that difference with the gene or DNA, that is also a genetic marker. Or now, as we have advanced sequencing technology, we could sequence the DNA and we can compare the sequences of two different individuals or two different species. Then we can say if they are, how far they are different from each other with respect to the DNA sequence. Whether these two species or two individuals are having the identical sequences at a particular fragment, or they are different. So broadly, these three things we can call it as a genetic markers. Now coming to the DNA markers. So here, you can see here, the names of three people has given here. One is Annam Pavan Kumar, Annam Shiva Kumar, Annam Mohan Kumar. Among these three names, you could observe that the initial name is common among these three people, and the last name also common among these three people. So that means this region is conserved among these three people's name, whereas the middle name is unique. It is different. It is specifying a particular person. So now, to identify a particular person, if I if I say what is your name, if three people have gone to a party and someone has asked what is your name, they said that Annam Kumar 1, Annam Kumar 2, Annam Kumar 3. It doesn't make difference. I mean, it's very difficult to identify the person. So if they say their middle name, then it is easy to identify who is he, who is who. So then when we apply the same analogy to the DNA, in the when we are comparing the two individuals or two different species across in the genome, there are certain regions which are conserved across the species or across the individuals, whereas the region between these two flanking region, you can see in the blue box. So the blue box regions are conserved across the species, whereas the black color or red color box sequences are there. They are differing between the species. So if we design the primers for this region, if we are targeting these two regions, and then we can identify what is the difference between the primer binding region. So altogether, this entire segment is known as a marker. So a marker consists of conserved region as well as a variable region. So like that, in the entire genome, there are different markers are there, and these markers have conserved region as well as the variable region. Conserved regions we use to locate at that particular gene at that particular location, and the variable region we use to see whether they are how much variation is there, whether they are similar or whether they are different. If they are different, we can quantify the variation, and then by using the statistical tools, we can estimate whether they are the same species or different species, or the same population or different population. So why the sequences are conserved across the individuals, across the species? If you see the tree of life, we all have evolved from a single common ancestor. So there is a uniformity across the life. So those uniformity is manifested in the form of the conserved region. So that is the beauty of the DNA marker. So we are searching for the conserved region, and also by using those conserved region, we are looking at the region between the conserved region and to find out the variation. So this is a broad idea, the core idea. Marker means it should have the conserved region as well as the variable region. So we will see one by one the very brief overview. So RIPD, in the case of a random amplification of polymorphic DNA. So this is the basic and a primitive marker technology where the conserved region is primer binding sites. The primer binding sites are conserved, and the variable region are the region between the primer binding sites. If you see here, this is individual A and this is individual B. In individual A, the primers are, this is a one forward primer and this is the reverse primer. Here you can see primers are binding at 3 regions and they got the 3 amplicons here. Whereas in the primer B, they lost the primer binding sites here. So that's why you can see the 2 bands only. So here the conserved is primer binding sites, and the variable regions are a region between the primer binding sites. Sometimes the primer binding itself is a variable. So that is the beauty of the RFPD. In the case of RFPD, we don't require any prior genetic information. We use a very short oligonucleotides of 10 base pairs, where we just to get an idea of how much genetic variation is there in the population or between a species or within a species. Coming to the another marker, restricted RFLP, restriction fragment length polymorphism. So here the conserved, here we will use the restriction enzymes. So these restriction enzymes identify the specific RE sites. At the specific RE sites, they will cleave the DNA. And when they cleave the DNA, they will generate the fragments. So here the conserved region is the RE sites. The RE sites will be identical across the species. If you take the Katla, if you take the Labirohita, if you take the Sirahannes progala, so since they have evolved from the common ancestor, so they will be having the identical sites at that identical location. So when we use these RE sites, they will cleave that RE site at the RE sites, and they will give the different fragments. And those fragments, when we look at the grass gel, we can see the differentiation. Now coming to the microsatellites, in the microsatellites, the conserved region is the primary binding sites, and the variable region is the repeats between the primary binding sites. So here you can see in one individual, the amplicon site is 146 base pair, whereas another individual it may be around 145. So now coming to the another marker, that is a single nucleotide polymorphism. So this is the latest markers which people have most widely used in the single nucleotide polymorphism. So only one nucleotide will be changing across the individuals or across the species, and whereas the flanking region, the sequences surrounding this polymorphic region will be conserved. So these SNPs are, since they are located at the coding region, so they will change the proteins, and they will responsible for the trait evolution. They are responsible for the adaptive variation. So now, because of the sequencing technologies evolution, so we are capable of sequence the genomes, and we are capable of scan the sequences to identify the SNPs. So these are all the broad markers like RAPD, and people are not using, because there is a low reproducibility, but it will give a brief idea about how much genetic variation is there. So whereas the RFLP and SSRs, that is the simple sequence repeats, and microsatellites and SNPs are are very good in nowadays. So if you see the evolution of the molecular markers at the AFP stage, people were using the allozymes, and then restriction fragment length polymorphisms and mini satellites and microsatellites. With the advent of the RAPD as or PCR, the RAPD has taken during the 2000 to 2003, and then AFLP amplified fragment length polymorphisms and single nucleotide polymorphisms. But nowadays, the SNPs and the microsatellites are most widely used markers for different for addressing the different biological questions related to the fisheries science. Now coming to our topic today. So now I think it is clear that what is the DNA marker. So DNA marker is a region in the genome, in the DNA, where it has the conserved region as well as the variable region. So by using the conserved region, we are trying to estimate the variation among the individuals and among the species. And why we are trying to estimate the variation? Because the genetic variation is the raw material for any species or any life organisms based upon which it could evolve. So we can, it has several mechanisms, it has several advantages. If we quantify the genetic variation, we could use it for several purposes. So that is coming to the sustainable management of the fisheries resources. So the first and foremost thing is that for sustainability means it's a perpetual use, and which the resources should be available for forever, and at the same time it should give to we can exploit them, and we can use them. At the same time, the resources are available for future generation. So for that, we have to assess the stocks. What is the stock status, whether it is depleting or whether it is enhancing, or it is depleting, what is the mortality rate, what is the stock recruitment rate. So before that, we should be able to identify what is the species. If we are able to identify the species, then only we can assess the stock, and then only we can formulate the resource management measures. So if we see the flow of the processes required for the sustainable management of resources, one is the species identifications and the fisheries. Once we identify the species, and then we can assess the stock, and then we can resolve the mixed stock structure, and we can assess the stock recruitment, and we can assess the ecosystem monitoring, and we can detect the invasive species, and we can finally stop the illegal, unreported, unregulated fishing. So among all these processes, DNA markers have a tremendous role to assist the fishery managers for sustainable management. Now coming to the first one, the species identification. So why we need to identify the species? So if you see the ecology, if you see the life, the functional unit of the life is a cell. So how a cell is acting, based upon that, the individual will also determine how the individual respond to the environment. If you see the ecology, the functional and the fundamental unit of the ecology is the species. The species And the species of the individuals of the species, they form the populations, and the populations, they form the communities, and the communities form the ecosystems, and finally biome. So if the species is in a healthy conditions, if the species genetic diversity is at the healthy conditions, so we can we can protect the populations and community and overall the biome. So that's why identification of the species is very, very important for the resource management. And also the ecological balance. If you see the coral, if you see this picture, there are so many fishes are inhabiting the same ecosystem. So it doesn't mean that some of the fishes are having the different, very important role, and some of them are not having. Each and every species of this ecosystem have a unique physiology, and they are having the equal and very equally important role in the ecological balance. So that's why each and every species has a right to stay in the ecosystem, and each and every species has to be characterized, and it has to be protected for ecological system, ecological balance. And finally, since they are giving the services to the humankind, like which starting from the medicines and plants and everything, is we are depending upon the biodiversity, and without biodiversity, the humankind would have wiped out very early along. So with this background, coming to the philosophy behind the species identity. Correct. If you see this one, we can see the color like this combination, whereas the puppies, they see the colors in a different, they may not, in a different way. And even if you see the petals here, we can see the only one color, whereas the insects, they could see the two different colors. So here the point is that what is the reality actually in reality, whether it is what we are perceiving the things, whether that is the reality, whether other animals, how they are perceiving, that is the reality. So in real sense, the reality, knowing the actual reality is a very difficult task, and it is almost sometimes it's impossible. But we are using the different tools to observe the reality, and by using those tools, we are trying to understand that phenomena, and we are trying to understand those concepts. So here, suppose imagine here one phenomenon is there. One scientist, he has proposed a theory, one theory. Based upon his theory, he is trying to understand this phenomena, and based upon his theory, he is understanding this reality. Both are correct and both are not correct. We may say that. So it is depends upon the how logically they have formulated their perception and how logically they are putting their ideas. So one kind of that is the species. The species also, the concept of the species itself is the most debatable question, and it is still so many people are asking how to define the species. So even though we have the different definition of the species, but the operational definition is very difficult. So biological species concept, here one scientist, he observed these two species, he could say as per the biological species concept, he can say that there are two different species. But when the another scientist, when he applied the morphological species concept, since they are appearing the similar in morphology, so he could say the same species. So here your logic and your hypothesis and your theory determines your theory addressed to understand the reality in this case of the species. So like that, there are so many critical thinking brains. Each brain has its own theory, and they have their own logical things, and they put their things in a logical manner and proposed so many species concept. So I'm not going to explain all these things, but this is the concept. So now morphological species concept, there are certain based upon the morphological similarity, we could take it cluster the individuals into one entity or one species, but still it has some limitations, like because of the phenotypic plasticity. So though they are the different species, because of the environment, so they should reflect the similar morphological characters because of the convergent evolution. So it could lead us to name them as the same species, although genetically they are two different species. So this kind of, here you can see if the same species is in a different environment, so they could reflect, their phenotype will be different. So if we go with the only phenotype, we might say that probably there are three different species. Just I put this slide for your understanding. So what I mean to say that the phenotype may sometimes deceptive. So we need to use the molecular markers where we could see the variation at the genetic level, and based upon that, quantifying based upon the threshold of the genetic variation, we could cluster the individuals into species. So one more thing, another example is that morphologically, these two species individuals are looking so similar, but they are two different species. So these are some limitations of the traditional taxonomical tools, and alternative tools over the years. So many methods have been developed as the science is progressing. That is from the chemotaxonomy, where the molecules will be compared among individuals, and the cytotaxonomy, where the karyotyping will be observed, and now the DNA-based taxonomy, where the DNA sequences are aligned and compared, and based upon the similarities, a threshold value, a divergence, distance values have been proposed, and based on those values, the individuals are now grouping as a different taxonomic individuals or taxonomic units. So basically, this is the logic behind the genetic distance molecular markers. We will be using the phylogenetic species concept. The phylogenetic species concept, the monophyletic DNA. So we will observe the individuals of the same species. They form the monophyletic clades, and those individuals, we will categorize them as we will say them the same species. So as I said previously, species are populations or group of populations that share the common evolutionary fate through time and over the time. So because of the evolutionary and because of the other biotic and non-biotic factors, the population may be split into two different subpopulations, and these subpopulations will be evolved over the time as two different species. So as per the phylogenetic species concept, the evolutionary lineages will be taken. Each evolutionary lineages will be considered as a different species, and they will form the reciprocal monophyletic in the phylogenetic tree. And based upon those clusters, we could say that these individuals are different species. So, oops, sorry. So coming to the basic idea behind the DNA-based taxonomy. So generally in morphological taxonomy, what we will see, we will estimate the morphological traits, morphomeristic traits, and we will compare so by using the statistical tools, we will see whether they are falling in the range or not. If they are not falling in the same within the range, so then we will describe them as two different species. The same way, what we will do in the case of DNA, we will sequence a particular fragment of DNA, and the particular fragment of DNA, we will compare their nucleotide sequences. If they are identical, they may be the same species. If they are not identical, based upon the dissimilarity, we will classify them into different species. So which gene for which DNA fragment? So there is a molecule in our body which determines the power, sex, and suicide. So powerhouse of the cell, that is the mitochondria, and the maternal inheritance, which determines the sex in some of the individuals, it is the mitochondria. And suicide, it has a programmed pathway which leads to the apoptosis. So a beautiful book written by Dick Lane on this with this title. So power, sex, and suicide, the mitochondria and the meaning of life. So this mitochondrial DNA has the maternal inheritance. That means except some of the muscles and most of the metazoans, most of the other vertebrates, the mitochondria follows from the maternal inheritance. And this, the variation which we could observe at this mitochondria is the evolutionary variations most of the time, but not at the population level between the species. So variation in polymorphism of DNA is inversely related proportion to the selection proportion, the pressure of the fragment. So whatever the fragment we are selecting, if we are observing high variation across the individuals, that means the fragment has a less selection pressure. So that's why it is evolving over the generation. So those kind of fragments are not useful for the species identification. So for species identification, we need to select a fragment which has the moderate selection pressure, which could attribute that variation with the speciation events. So that kind of fragments are available in the mitochondrial DNA. So that is the cytochrome C oxidase subunit 1 gene as well as the 16S ribosomal RNA gene. So these gene fragments we could use for the species identification, and we can say species delimitation, not identification. So briefly, I will explain this process that in the case of this process, we call it as now people have started it as a DNA barcoding concept, and the DNA barcoding concept, we will be using the cytochrome C-oxidase subunit 1 gene. So in this methodology, so we will be taking around 5-6 individuals from the different locations. When we fragment and when we amplify and sequence this fragment, you can see here within the CUTLA, so the variation out of 10 nucleotides, only one nucleotide is changing across these 5 individuals, 5 individuals. So within the species, the dissimilarity is only 1 nucleotide out of 10. So within the species distance value is 1 is differing out of 10, that is 0.1 distance value. So whereas in the ROHU, the same fragment has a different nucleotide sequences. When we compare within the species, out of 10 nucleotide, only 1 nucleotide is differing. So then within this ROHU, the distance value is 0.1. But when we compare the CUTLA and ROHU sequences together, so you can see the difference is 4 nucleotides are differing out of 10. So the distance value is 0.4. So the distance between within species and between species, so that is 0.3. We can call it as a barcoding gap, which could attribute to the evolutionary distance values. So if we see the basic science behind this one, the scientists have used the dating approaches, and using those dating methods, they identified that most of the species have 1 million year divergence time. So they have separated from their common ancestor with a 1 million year time frame. So then after for 1 billion year, the mitochondrial DNA will accumulate 2% of variation. That means out of 100 nucleotides, 2 nucleotides will be fixed in the species for a 1 million year time frame. So that means if we see the 2% divergence among the individuals, so it indirectly indicates these individuals or these populations might have separated from each other. There is no gene flow between these two populations over a 1 million year of time. So based upon that logic, based upon that 3%, now the people have, some of the species may be having 3% divergence. So that means 3 nucleotides are fixed in the species out of 100 nucleotides. So when we observe the 3% or 2% divergence among the individuals, so we can say that those individuals might belong to the 2 different species, not belong to the same species. So this is known as a molecular clock hypothesis. So as per the molecular clock hypothesis, the amount of genetic difference between the sequences is a function of time since their separation. If the sequences are having very less variation or very high similarity, that means they have not diverged from their common ancestor, that they are sharing the same gene pool or they are in the same population. If the variation or if the differences is very high, that means the individuals might have diverged from their common ancestor since a long time. So that's why they have accumulated much variation. So this is the proved, a molecular clock hypothesis, and based upon the molecular clock hypothesis, scientists have dated the molecular clock around the molecular divergence, molecular mutation at 2%. If 2% in some groups, most of the groups are as 3% is the threshold limit based up above which the individuals might belong to those different species. So here you can see this bar in this chart, the orange color peak represents the within species, and the yellow color or this one represents the between species distance. In the previous example, this is 0.1 and this is 0.4, and this gap 0.3, it represents the barcoding gap. So, but some of the species may not be having the barcoding gap, and that within species distance the values may be overlapping with the between species distance values. And in this process, the CY gene or barcoding approach may not be useful. So here the beauty of the barcoding, this CY gene sequencing for species identification is that irrespective of the geographical distance, you see in the x-axis, it is showing the geographical distance, and y-axis, it is showing the, you can see here, distance values. So here you can see within the 0 kilometers and 5,000 kilometers and 10,000 kilometers, if individuals of the same species, they have collected from their distant locations. However, the distance values are very low, less than 0.04. Whereas the different, this color shows the interspecies, a different species from the within the 10-kilometer radius or within the 5,000-kilometer radius. So irrespective of the geological locations, the genetic distance values between the species are always high. So that shows the beauty of this gene, that is the cytochrome C subunit 1. So this gene has the sufficient phylogenetic signal, and that signal can be tapped to segregate the individuals or to cluster the individuals as per their phylogenetic species concept into different species. So these are different modes of the speciations, allopatric and sympatric. In allopatric, dispersal, so where the individuals will be moved to another geographical locations, and after that they will lose their connection and the gene flow will be stopped, and over the period, these individuals, the subpopulations will be evolved with different species. The vicariants, whereas the tectonic plate movements, the individuals are in the same plate, and because of the tectonic plate movements, some of the continents, some of the land mass has been drifted away from this land, and over the time they have lost the gene flow and they evolved into different species. So the classical example is the Neotrigon Kubli complex, which is abundantly available in the Indo-Australian coastal shelf line. So here we previously thought that only one species, that is the Neotrigon Kubli, but the recent molecular tools have shown that there are different species which could be, there are different lineages which could be elevated into species rank. And sympatric speciation, they are in the same ecosystem, but due to some heterological parameters and biological parameters, the gene flow is stopped and they would not share the gene pool, and slowly they will be evolved into different species. So the classical example is the Tanzania lake from the South Africa, so where the different cichlids, so they evolved into the same lake. So now in this species identification, there are two processes. One is the reference database and application phase. In the reference database, it is the same as the Aadhaar card principle, where the people will go to the Aadhaar card station and they give their biometric data, and this biometric data will be tagged with the individual's data. So then finally the Aadhaar card will be issued like this, and where if you scan this barcode, and you will get the individual details of all this in the human being. So in this the same way, the DNA barcode, so we have to depend upon the classical taxonomist, and based upon the classical, with the inputs from the classical taxonomist, we will identify the species and we will extract the DNA of and then amplify the CY gene, and we sequence the CY gene, and each nucleotide will be given a different unique color, and this sequence will be considered as a barcode tag for this particular species. So all these sequence barcodes will be deposited in a public database, that is the barcode of life data system. So these bold systems have the curated data, and the most of the wrong nucleotides will be curated and they will be validated using the good quality sequences. So it shows the correct sequence repositories and specimen repositories and which labs, how many have been submitted. So from our lab, we have developed the barcodes for cephalopods and LS ranks and verbetes and the ornamental fishes and the mollusks and some of the peciforms and some of the pathogens and some of the LS teleos also. So now coming to the application phase, once we have the reference library in place, so which is a curated reference database, so it has a tremendous applications in fisheries resource management. So here what we will be doing, so if someone has given a tissue sample, so because the larval stage and the processed stage, it is very difficult to identify the species as it doesn't have any morphological tools. So in this case, from the tissue or from the processed sample, we will be isolating the DNA, amplify the CY gene and sequence, and the sequence we will submit to the database, and we will see the sequence similarities, and based upon the sequence similarity, we will identify what species it is. So here we have submitted the sequence of the shark fin, and we have seen that the submitted sequence has matched with Spirna liveni, 100% similarity. So here we could say that the ray fin is actually from the Spirna liveni with 100% confidentiality, we can say. But again, here it depends upon the accuracy of the database. If the database is much more accurate, we can identify the unknown species. So we have identified some mislabeling in the Indian coast, where most of the fillets were mislabeled with the fungaceous fish. And also the ready-to-cook fish also, products also labeled with the low-value fish like Oripanachapuras, and some of the ready-to-eat products which were labeled as high-value fish were actually substituted with the low-value fish like Pangasiodon. So this kind of study we have published in Food Control, which got the national and media attention. And also we have verified the crab mislabeling. So now coming to the larvae identification. So most of the mangroves are the actually nursery and breeding grounds for the larvae production. But how many fish species are using the mangroves as their larval or breeding ground was not known because of the lacking the morphological characters of the larvae. So we have used the DNA barcoding approach and from the Maharashtra Mumbai coast, and we identified around the species from 13 important families for using this larvae as these mangroves as nursery grounds, and it was published in Journal of Coastal Research. And also sometimes ecotypes, even though the individuals may be slightly different, it doesn't mean that they are different species. So by studying the DNA sequences, that is cytochrome oxidase 1, we identified that these two species, we identified some ecotypes or morphotypes in Seripina fasa. So that also we have published here, and describing new species. And we have identified previously the new dragon flea from the Indian coast was considered as a new dragon flea, but based upon the polymer DNA barcode and other molecular tools, we identified this is actually the new dragon flea. The type location is the New Caledonia, but whatever the new dragon species are there, they are the different species which we have named it as a new dragon indica. So based upon the Indian Ocean. And also we identified a new teleos, that is the Stolliferus thameliensis from the anchovy, this is a small anchovies from the Tamil Nadu coast. And we also identified a new blind eel from the Mumbai, from a well of the Mumbai, that is labeled as Ractomyctis mumbai. And also we have identified a parasite, that is a Ellipsomyxa fungal parasite, Ellipsomyxa polyophthalmia from the mud skipper fish. So here we have used the COI gene and 28S ribosomal RNA to distinguish the individuals. So life always look for the betterment. Wherever the conditions are conducive, the life will extend its distribution. So likewise, we have used this barcode approach to identify the extended distribution of the different fish. So here we have observed the extended distribution of Macrobrachium indicum, and another goby fish, Amblygobius farina, in the Indian water, in another extended distribution or first record of the different waters. So this is the one thing for the species identification. So species identification, the COI gene alone is not sufficient. It also has some drawbacks. If it is a hybrid species, identification is sometimes difficult, and it is also a newly recently evolved species. That time also the COI gene may not show that much variation. So there are certain drawbacks. However, it has certain prospects and advantages in this species identification and also the authentication of the seafood products. So now coming to the stock identification. The stock, once we have identified the species, what is the status of its population, whether its population is having the single stock or multiple stocks. Again, the stock is also, the concept of stock also much debatable, because there are different concepts are there and different logical theories are there. So however, when I go with the genetic terminologies, so the genetic coherence of the individuals, I can say it is a genetic stocks. Those genetic stocks have the evolutionary potential, and they should be treated as a distinct evolutionary units, and they should be protected. So whereas if we go with the morphological tools, here you can see the fish throughout the Indian coast, they morphologically they are appearing similar, but genetically they could be distinct. If we use the molecular markers, we could identify the genetic variation across these stocks. Accordingly, we can manage those stocks, and we can conserve the evolutionary potential of these stocks for future resilience against the climate change. So for this, we have developed the microsatellites markers and mitochondrial DNA markers. So AFLPs and a single nucleotide. AFLPs for previously, it is using, but it's not that much, whereas the single nucleotide polymorphisms are the most widely used. So in our lab, we have developed the markers for the cookie stream, that is the Parapinepsis stylifera, and we have collected the stocks throughout the west coast and east coast, and we found that there is no much difference at whatever the loci we have selected. So based upon those loci, based upon those markers, we we reported it as a single management unit, single stock and single population, and the single management strategies measurements are sufficient. However, here the limitation is that we have only used maybe 10 markers. We have not used much more SNP markers. If we use that one, probably we get more resolution among the stocks and much more information. And the same way, we have identified the different stocks and distinct stocks in the case of the freshwater giant river catfish Sperata singala using microsatellite markers. So we have identified the 5 distinct genetic stocks of 5 distinct populations in the Indian river systems. And we also developed the microsatellite markers for the Lamnitis marginalis, and we are characterizing the genetic diversity in this freshwater pearl producing mussel. And by using those markers, by using those variations stocks, we will be using those stocks for selective breeding for producing the better pearl performing individuals. So for future prospects of these markers for stock assessment are using the SNPs. Most widely people are using the SNPs and using those SNPs to characterize the adaptive variation and fine scale structuring of the stocks across the Indian coast. So then third thing is that the age determination. How can we determine the age of the fish? Generally we use the otoliths, sometimes we will use the scales. So in the genome, there are certain regions are there which could act as a clock-like markers. So those markers are telomeres at the end, at the edge of the chromosomes. The region is called as telomere. With the age, these telomeres will be shrinking. So people have recently people have started using these region as a marker for estimating the age. So they have got different results. So some of them has got the positive correlation with the age, and some of them has got nothing, no correlation with the age, the telomeres length. However, it has a good prospects in the future, which could be developed as a novel markers for age determination of the fish. And ecosystem monitoring. So now the ecosystem-based fisheries management is an upcoming and emerging area where the entire ecosystem will be treated as a unit, and each and every species has to be protected, and each and every species has to be assessed its status, its conservation status, and everything. So that case, each and every prey to predatory, every species has to be documented, and after documentation, you could manage the things in a sustainable manner. So here for the ecosystem monitoring, we can use the eDNA, that is the environmental DNA. So most of the individuals in the ecosystem, they shed the dead cells and they shed the excreta. Through that, the DNA will be released into the water. And by taking those water without harming the individuals, we can identify the new technology, new approach is there, that is a meta barcoding. The meta barcoding is the extended form of the DNA barcoding, where a pool of the DNA sequences will be amplified and sequenced using the next generation sequencing technologies. So here you can see the eDNA, it has a different DNA molecules from the different organisms. So each DNA will be amplified using the next generation sequencing methodology. And by using the bioinformatics, we can cluster the sequences as per the OTUs, and then we can classify or identify how many species are there in the ecosystem, and then we can frame the food chain mechanism, which is predatory, by using the different models, simulation models, and we can understand the ecosystem better way. And then detection of invasive species. Now it is so rampant in the Indian waters. Wherever you will go, you will find there that is a Pterygopteryx pardalis or Pterygopteryx juvens are these hybrids. So by using the eDNA and by using the eDNA, we can detect. The early detection is very important for preventing the invasive species. So for preventing the invasive species, for surveillance of the invasive species, the eDNA and the amplification of this cytochrome oxidase one other markers so will be useful for early detection and timely control of the invasive species. So apart from that, there are other applications like the estimation of the harvest rates and abundance. So we can use the microsatellite markers to estimate how many individuals have been harvested, what is their abundance. So it requires the creation of the database and also then identifying the allele frequency and matching the allele frequency with the database, and also the genetic effect of stock enhancement, whether really the effective population size has been increased after the sea ranching or not. So all these genetic parameters will be estimated by using the DNA markers, and these could be used for formulating the better management measures and sustainable resource management. So with this, DNA markers have very prominent and very important role in each and every aspect of resource management. So with this, I conclude my session. Thank you.
Thank you, Dr. Pavan Kumar, for delivering this wonderful talk, excellent talk, and I enjoyed a lot, learned a lot, new stuff. Even for an economist, I could follow the talk, and I'm really appreciative of that. Before going to the discussion or leave the party, we provide you a feedback link where you can give us a feedback about today's. So the link is in the chat box. We would also like to announce if one of you are happy to volunteer for AFS Talks, please do let us know. We will be interested to know in which ways you are willing to contribute developing our AFS Talks in a different level, in a higher level. So for example, if you like to hear if you have like a website development skills, conducting, developing skills, training and assessment skills, there are a host of activities that we are looking forward to deliver through AFS Talk. So please don't hesitate to contact us and discuss what value that you can bring in to AFS Talks. Now, we can move on to the discussion part. We have 15 minutes of discussion. Feel free to unmute yourself and ask questions to the speaker from this point onwards. If possible, you can also type the question in the chat box so everyone, including the speaker, can read and understand the question. Thank you.
Thank you, Dr. Pratisha.
Okay, I saw a couple of chat. Seems like it's not a question, so they are saying that a very interesting and informative talk. Please kindly upload in the AFS YouTube channel. Excellent talk, Dr. Pavan Kumar by Shine Anand, and Chanakya Naidu said that very thanks for the interesting and informative presentation concept.
Like, can I ask one question?
Yes, sure. Deepak.
I know. So very interesting talk. You try to cover a lot of subject in short time. So anyway, so I think you explained everything that you could possibly do with DNA markers. The one that I was not much aware is about the aging, like how you can find age of a fish, and that looks pretty interesting to me. And I just want to know a little bit more about it, as well as a key concern I feel is like, wouldn't be that a more expensive way of finding age than the traditional methods? And when you consider that, how much accuracy can we expect from that sort of analysis?
Yeah. Good question, Deepak. And so here, actually, the baseline data, like if the telomere size, which region of the telomere has to be standardized. Suppose if it is a juvenile and sub-adult and adult and other stages. So if we have the baseline data about this is the telomere size, length of the telomere at the juvenile stage, and the length has been shrinking over while it is aging. So from the Australia, Queensland University, Dr. Ovenden, so she was working on this area, and they got some success, but at the same time, there was not much correlation statistically. So still people are looking for other regions where they can, but in humans, it was proved that as we are aging, the telomere will be shrinking. So it has proven that that one. But in the case of coming to the fishes, it is economically at current point of time, it might not be feasible, but over the time, as the sequencing technology has been reduced and the analytical methods are being developing. So definitely it could be an alternative method for the non-invasive method for age determination. So it's still in an infant stage.
Thanks, Pavan. Yeah, when you get a chance, can you send me the paper just for reference?
Sure.
Yeah, thanks.
Dr. Pavan, there's another question on the chat box by Shilpa. Shilpa, do you want to unmute yourself and ask the question?
Yeah, can you explain? I can read the question. Can you explain the concept of subspecies? So subspecies, it's a when it comes to the molecular tools, it is a tricky question. It is a tricky thing to define. It is a subspecies based upon only the molecular tools, only based upon the molecular genetic distance values. So because it has not been defined, this is the threshold value. If you get this much a threshold value, this is the subspecies. So it is in this case you require the comprehensive view from different morphological, osteological, and other traits. So to name it as a subspecies. So here we have we can name up to species level, but subspecies there is a sorry from the molecular taxonomy point of view. Can we use CBOI as a molecular marker for stock ID? Yeah, Shekhar. So as I said that whenever which gene you are selecting, it depends upon which question you are going to address. COI gene, so in the mitochondria, we have different genes. COI gene has moderate selection pressure. So moderate selection pressure means a moderate variation only you will get. So the variation which you will get might mostly corresponds to the evolutionary variation, not the population variation. So that's why for population genetics, you should use the markers which have less selection pressure, more polymorphism, such as D-loop or such as microsatellites, nuclear markers, or sometimes the cytochrome B also. Even though they are coding genes, some of the genes have high polymorphisms, which could be useful for the population genetics. But in the strict sense, D-loop and microsatellites are most useful for population genetics to get the clear picture of the stock structuring and variation. Balakrishna Chintada, molecular markers are mainly having the ecological aspect. If we see in industry, these tools can be very useful in identifying the fish products, especially for the high value fishes. Thank you.
Pradesh, because we are recording this, and can you read out the question for Pavan and then he can answer?
Sure, yeah. So just want to add to Dr. Balakrishna Chindara. I that was one of the things that I was thinking when you did the presentation. You show the slides where you show the products identify the fish species. Mislabeling is one of the biggest thing that seafood market is handling. So DNA markers can be useful in the sense of identifying those fish product and to control the mislabeling of the seafood products. I don't know how far that they are doing right now, but that's kind of a need of the industry.
Yeah.
So there's another question from Chanakya Naidu. So can environmental pollutants affect genetic makeup of organisms and thereafter results in new species?
Yeah, so there is a possibility. So that genetic, especially the pollutants, most of the time those pollutants cause the genotoxicity. So the genotoxicity means, so whatever it is happening, any modifications, any alterations are happening in the genetic material, that should happen in the gametes. So then only it will pass to the next generation. Then only it will become the heritable trait, and then only it will create a new allele. If it is happening in the somatic cells, so it might not lead to the new species, or it might not lead to the new allele. So speciation is a long-term process, and first a new allele, new variation has to be created, and new allele has to be created, and that allele has to be increased in the number in the population. So it takes a long term, but as you said, there is a possibility that the pollutants and the environment could induce the mutations in the gametes, and those gametes will create a new allele, and that allele Yeah, this it happens that allele might be resistant to the pollutant, and those alleles will subsequently increase in the population, and it could cause a new species or new in population that is resistant to that pollutant. So it happens. There's a possibility, but still there are certain rights are there. I mean, riders are there.
Okay. Hope Dr. Pavan answer your question, Chanakya. Moving on to the next question, Ashish asked, like, can we use eDNA approach for food web analysis? And also he asked another question also, can we tell location of a particular fish species by using eDNA technology?
Yeah. So for food web analysis, we can use the eDNA, but we will understand what kind of species diversity is there in the ecosystem. But again, you need to depend upon the traditional tools for prey and food, the gut content analysis, and what kind of food it is consuming, and to identify the prey and the predatory food systems. So for that food web analysis, the eDNA will say will reveal you what kind of species composition is there. And again, you need to depend upon the classical biological tools to understand the food web analysis. So eDNA is a tool which will give the inside information, but for getting the overall picture, again, you need to do some biological aspects of that particular fish. And the second question is that a location of a particular fish species by using eDNA technology. So for this, we can use the SMP markers or microsatellite markers, and we need to develop a series of markers, a database, and those markers are stock specific, and we need to define the geographical periphery or the geographical expansion of that stock. And suppose if I say that this marker is available in the Mumbai stock, and we should know the how far the Mumbai stock fish is capable of migrating and capable of moving. So we need to know the distributional range, and we need to have a series of markers, and we need to have a strong database, then it could be possible. So now the traceability is a very big, important issue for the sustainable fisheries. So this is a future area where we can work on.
Okay. So we have another question from Shekhar asking for how D-loop works.
So D-loop in the mitochondrial genome, you see only the D-loop which doesn't code for any protein. It doesn't mean that it doesn't have selection pressure. It might be having a role in the replication, but compared to other genes, the D-loop is not coding for any protein. So hence it has the less selection pressure, and you will get more polymorphism, and that polymorphism we can contribute with the population.
Okay. Suresh Babu has asked one question. Can we use a formalin preserved fish for barcode?
Yeah, so previously it was difficult to get the DNA from the formalin, but now with the advent of different chemistry and improvement in the chemistry and other electrolytes. So now the new kits are available. So especially from the mole bio and Kaizen. So from even the museum specimens also, people are extracting the DNA archived specimens, and now it is possible, but the success rate is not as we expected, but it is possible with the advanced available technologies, available kits. Sorry.
Thank you. And Ashish is asking one more question. Can you tell the most important Indian paper which is related to eDNA?
So unfortunately, in the Indian context, the research on eDNA is very, very, very minimal. Only recently one paper from the National NCSM, National Center for Sustainable Management, Coastal Sustainable Management. So they have published one paper, and that is a brief descriptive paper, but not much work has been done in the case of eDNA. So we are expecting a few more papers from our labs in the future.
Okay, thank you. Shekhar is asking which software can we use for stock ID?
So it depends upon the kind of markers which are using. So there are different softwares are there, and if you are using the microsatellites, I mean, it's not like which software will give you the answer. It depends upon the which genetic parameters you are estimating. So based upon the genetic parameters estimating, so you can select the appropriate. There are so many softwares are there which will estimate the genetic parameters, and based upon the genetic parameters, you can say whether they are single or multiple stocks. So I cannot say that, but there are several softwares are there like the POP, gene POP, DNA sequence polymorphisms, and n number of Harlequin is there. Yeah.
Okay. Thank you. And Ashish was asking who used the, I guess it's related to the eDNA question. Who used this one first time in India?
It depends upon the resources which you have. So if you have eDNA, it requires a huge funding, and if you have a huge fund and you can procure the kits and you can order the NGS method, and since it's in infant stage, only even the descriptive experiment also will publish in the paper. So there, we cannot say this is the first, he is the first person. So people are working in the different labs, and so I have seen the paper from the National NCSCM Chennai.
Okay. Thank you. At this point, we don't have any more questions on the chat box. If anyone at this point want to ask any question, please unmute yourself and then ask directly to the speaker. I saw Deepak has raised a hand. Deepak, is that from your earlier question, or do you have a new one?
I have a new one.
Okay, go ahead.
So, Pavan, you briefly went through the DNA marker application in the growth and recruitment in the fish stock. I'm a fish stocks assessment guy, so I'm just more interested in that area. Can you explain a little bit more how these markers can be used for that purpose?
Yeah, so for this Deepak, we need to have the, as I said that several times, the baseline data. Suppose for the stock, the traditional method, what we do, mark, capture, and recapture. So that's so that kind of method is there for estimating the harvest rate and abundance. So here, instead of the physical specimen, so we develop a series of molecular markers, and we estimate the allele frequency of the particular stock, and then we got the catch the next time. So we will compare the allele frequency with that fish, and then we will use the statistical method to know whether it is whether that particular individuals are abundant in the stock or not. So that could take a different. I mean, it could take an entire time to explain it more precisely.
Did you say allele frequency?
Yeah, allele frequency. So suppose, yeah, we can develop a series of markers, and for that series of markers, for each marker, we should estimate the allele frequencies of the particular stock. And again, when we capture that one, so we will compare whether the allele is falling in that frequency or not. So based upon those methods, we could say whether the harvest rate is more or abundance is more.
Yeah, can I bring you in for that talk? It's a fun one. You have a paper on that stuff, or
we are not working on that area. We require some, I mean, a little more fund. That is the problem, and we want a genetic analyzer for that purpose.
Oh, okay.
So, so the only thing is that funding. If we have fund, we can do a lot of research. If we don't have fund, then we do simple barcoding.
It's a reality, right?
Yeah.
All right. Any other question? I saw Vijay had some emojis up. Is that? Do you have a question, Vijay? Or if it is, like, please unmute yourself and then ask the question. Okay, I don't think. Okay, so he doesn't have a question, so I think it's just an emoji that he put there. All right, so so it brings the end of all the questions. Thank you again, Dr. Pavan Kumar, for such an excellent talk. Like I said, I'm an economist. Like I I have gone through some of these when I was like, doing my master's, and this time it was like kind of a refresher one. And then I honestly, I learned a lot today. So thank you so much. And I guess most of you in the chat box from the comment from most of the listeners, I feel like, it's an interesting talk for most of those as well.
Can I request everyone to just provide a feedback? The link is
Yeah.
there in the chat box. Thanks.
Yeah. Thank you. Thank you again. Thank you for attending this AFS talk, and you guys all have a wonderful rest of your day, and we'll see you in the next talk. Yeah.
Yeah, thank you. Thanks for all the audience. I mean, despite I was so worried whether people will be stay together or they will move, but I'm happy to see that all are together. And thanks, it gives it will give motivation for to me for further subsequent talks and wherever it is possible. Thanks to Deepak and thanks to Dr. Pratisha and for giving me this opportunity. Thank you all. Thank you. Yeah.
And Pavan, send your students next time. We want more people to listen to talks.
Yeah. Sure. Thank you.